Welcome back to our channel BioLab, and this is VAMSI. In today's video we will be starting the physiological effects of PGRs. In yesterday's video we have seen the introduction and discovery of PGRs. Today we will be seeing the physiological effects of PGRs, and we will be mainly focusing on the physiological effects of auxins, our very best friends auxins. These are PGPs or PGIs. Think one time they are PGPs. Yes. In yesterday's video I have told you about what are PGPs. They are plant growth promoters. They are involved in the growth promotion activities. Yes, if you haven't watched the yesterday's video, please go and watch it now. It would be in the playlist. You can go there and watch it. Yes, our next lecture would be from your zoology part, and that would be also from your human physiology on locomotion, and the notes I will be uploading it soon on the website. There you can go and check it how to get the notes. Yes guys, so without any further ado, let's get into today's video. Today we will be seeing the physiological effects of auxins. We have some introductory points about auxins, and you no need to write this. I will be saying when you have to write them. Auxins. Write the first point, it is very important. First 4 points you have to write compulsory. These auxins are basically the derivatives of indole acetic acid. They are indole acetic acids. You can also call them IAA, indole acetic acid. Auxins are indole acetic acid compounds. Remember this point, please write. Auxins are the derivatives of indole acetic acid, and they are also called as IAA. Yes, hope you have noted it. Now, auxins were first isolated from let's take white. Yes, auxins. These were first isolated from human urine. Remember this point, very important. Urine. In plants, they were first isolated by FW vent and oat seedlings and the coleoptiles of oat seedlings, and they were first isolated from human urine. Remember this very important point, please make a note of this too. Auxins were first isolated from human urine. Hope you have noted. I am removing these points. Let's move on to the second point. Today's lecture will be all of points only. You have to note them, but at some places you can relax in today's lesson. Yes. Let's continue to the next point. These auxins can be of two types. Yeah, something fishy. Yes, no need to write these types. You have to just remember the examples. Then there can be natural auxins. There can also be synthetic auxins. Natural and synthetic auxins. You have to remember the examples, very important. There can be too many questions on these. Yes, let's write what are natural auxins. These are naturally occurring auxins. As per the name you can see, they are naturally occurring auxins. They are synthetically prepared in the laboratories. Now, naturally occurring auxins examples are, remember these, as we have spoke before, IAA, indole acetic acid. It is also an oxin, and oxins are the derivatives of indole acetic acid. Yes, IAA, indole acetic acid, and IBA. Remember these two. Only you will be having two examples each. IBA. What is this IBA? Indole butyric acid. Indole butyric acid. So now here I have a question from the previous chapter. Let's see how many could recall the previous chapter which we have taught. Yes. From where is this BA prepared, butyric acid? From using which microbe is this prepared in the industrial uses? Yes, Clostridium butyricum. Yes, Clostridium butyricum, butyric acid. It's just a small question from the previous chapter, not related to this. You no need to worry. If you haven't watched that on microbes, go there and check the playlist and watch all the videos. Let's go. IAA, indole acetic acid, IBA, indole butyric acid. Now the examples of synthetic oxins are, here also there are two. NAA, naphthalene acetic acid, or you can just remember it as NAA, and also 2,4-D. This 2,4-D, yes, hope you have heard it somewhere or read it somewhere. Yes, 2,4-D, it is an herbicide. Let me write NAA. Remember, let me write the 2,4-D first of all, since we have explanation on it. Example 2,4-D. Remember this, that is an herbicide. It is toxic to only dicots. It is toxic to only dicots, not for monocots. It will not kill monocots. It will only kill dicots. Okay, it will only kill dicots. Remember that, it will only kill dicots, and that is 2,4-D. It is also called as weedicide or herbicide. Remember that. There can be a questions. Yes. Now no need to write, only write these examples, put natural oxen side heading, synthetic side heading, write IAA, IBA, NAA and 2,4-D. That's enough. Now let's move on to this last point, and then after we will be seeing the physiological effects of oxins. This last point, before I tell you, you can just think of it. Auxins are growth promoting. Auxins help in elongation of the plant body, and from that you can take that auxins are present in the apices of root, shoot. Yeah, they are present in the apices of root and shoot. If this is, let me draw something which is, which I can explain. I have already shown you this drawing, but I am drawing it again. Yes, we thought this, the upper part till here it was a shoot, this lower part was root. Yes, this lower part was root, and here there are the presence of auxins. These auxins are present at these points, so they help in growth elongation. Okay, they help in growth elongation. Now let's move on to the physiological effects of auxins. Very important. Make note of each and everything which I am going to tell from now. If you have skipped the previous one, nonetheless, since we have the notes, and these are also in the notes, but please make a note so that they feed in your mind. They are very important, and for remembering it in the form of shortcut, it would be difficult for you for remembering everything. I'll be just saying you the important things. I will be underlining the important thing in a whole sentence which you have to remember. Just remember that. These auctions are helping in, they help in routing in stem cuttings. They help in routing in stem cuttings. Routing stem cuttings. Just remember that. Routing and stem cutting. See how, what are these routing, stem cutting, etc., etc. Yes, uh, in one lesson, in a lesson you have learnt about the methods for artificial vegetative propagation. Yes, in your 10th class you have learnt about that also. Methods for artificial vegetative propagation, and one of the method was grafting. Grafting. What is grafting? Grafting, it is a placing of one stem over the other one and tying it so that we tie it so that a flower of mixed variety would form, mixed color would form. So this grafting is mainly done in rows. Yes, did you remember that? Yes, these auctions help in this routing. If you place the stem of one over the other, they help in routing for nutrient transformation from this stem to the stem. So that's the role of auctions and stem cuttings. Okay, remember that. Routing, stem cuttings, and a fruit stem. Remember it. You would automatically get the point into your brain since you have options. Yes, in your boards you have to write the full statement, so you have to remember it. Yeah, in the NEET point of view, you know it. Yes, let's move further. The next point is they promote flowering. The very essential point. Promote flowering. In what? In some plants like pineapples. This is very important. Pineapples. In pineapples, generally, the flower appears little later. So, in order to get the fruit in advance, when we spray these auxins, they help in flowering in pineapples. They help in speed. They increase the speed of flowering in pineapples, and there you get fruits. There from that you get fruits very fast. Remember this: flowering pineapples. Yes, first I have said that root stem cuttings, flowering pineapples. Now let's see the third point of auxins. Yes, see. Now they prevent fruit and leaf drop at early ages. They prevent fruit, I'm just drawing it symbolically, and leaf drop. They prevent fruit and leaf drop during the early ages. Means they delay senescence. See, sometimes when you see some plants, their fruits and leaves start dropping when the plant is too young. So these auxins prevent that, and they promote abscission. What is this abscission now? Very easy definition. They promote abscission. It is a dropping of mature leaves and fruits which are close to senescence, which are close to oldness, those leaves and fruits are removed by these auxins. They promote abscission, the removal of the removal of mature fruits and leaves naturally by the plant is called as abscission, and that is promoted by auxins. Okay, they prevent abscission during the early ages, they promote abscission during the old age, during the mature age. Yes, remember that. Before explaining you the fourth point, I have some important definition that I have to tell you about apical dominance. And these auxins promote apical dominance. What is meant by apical dominance? Apical bud. Have you ever remembered it, or have you ever heard the word? Yes, these buds are present in the apex of the shoot. The buds which are present in the apex of the shoot. If this is the plant and if this is the apex, and these buds which are present at the apex are called as apical buds, and the buds which arise laterally from the branches are called as lateral buds or axial buds. Okay, axial buds. Now what happens is, if this apical bud is there, this apical bud in the presence of auxins would not let these lateral buds to grow. It inhibits the growth of lateral buds, and it helps the plant to grow only in this manner, only like this. On the axial part, only on the axis the plant grows, but on the width the plant does not grow if there are apical buds. That is called as apical dominance. The inhibition of the growth of lateral buds by the apical bud is called as apical dominance, and that is promoted by auxins. And if you remove this apical bud, successfully the plant will grow bushy, since the lateral branches grow from the lateral buds, and this is used in tea plantations. Please remember, it is used extensively in tea plantations. Very important, it is used extensively in tea plantations. Remember that. Yes. Now let's move on to the next function, last but one function, that is the fifth function. Yes, hope you are getting everything, hope you are remembering it. Yes, let's move on to the fifth function. These auxins induce parthenocarpy in tomatoes. What is meant by this parthenocarpy? The tomatoes which are produced from parthenocarpy, using parthenocarpy, those are called as parthenocarpic fruits, and they are seedless, meaning infertile, meaning not fertilized. Yes, the fruit would get matured without fertilizing, and therefore it would not result in seeds. So remember, parthenocarpy, just the sake, just remember seedless. For seedless, just remember seedless. They induce parthenocarpy in tomatoes. They induce parthenocarpy in tomatoes. Now the last function, sixth function. Yes, not your function, bros. You have concentrate here. There is no any marriage going on. Let's move on to the last function. They help in xylem differentiation. They help in xylem differentiation. Auxins help in xylem differentiation. Means they help in maturation of the immature cells, like we have spoke about meristems, to form into xylem. Okay, and yes, 2,4-D, which is used as an herbicide. So that's it for this video and the physiological effects of auxins. In the next video we'll be talking about gibberellins and cytokinins. So thank you guys. If you like the video, please hit that like button very hard and subscribe to our channel BioLab. Oh sorry, what happened? Yeah, thank you guys.